really happy to be here to present the most recent work that I have been working at Caltech in the Chi Lab. My work aims to understand how cells make developmental decisions in mouse and human development. I divided my talk in different topics. I'm going to talk about my background and professional experience, the activities I want to be part of UCSD, and then the two main projects that I have been working on. And the first one is the spatial analysis of RNA distribution during early mouse embryogenesis. And the second one is notch signaling that regulates cell fate choices during human development. And I want to conclude my talk by talking about how I envision my lab and then the future directions that I want to take as a principal investigator. So I was born in Colombia. I think I was very fortunate to be able to be born in one of the most diverse countries in the world. That made me amazed about where this diversity is coming from since I was a kid. And I was fortunate to have parents that were supporting me all the way. So I was, since very early in my life, decided to become a biologist. So I did my undergrad and master's work in Colombia and focusing on orchid diversity. So I was a plant biologist there. So working there on evolution of orchids, I found that there are some patterns of that these special plants acquire during evolution, and I wanted to know further where all these special features were coming from. But being in Colombia, I didn't have opportunities to study really developmental biology the way that I wanted to study, and using model organisms as people use here in the US and doing all this molecular biology. So I decided to pursue my PhD at Northwestern University. And then during my graduate research, I proposed and tested a novel hypothesis for neural crest formation and really found the origin of these neural crest cells that are unique to vertebrates and that have the potential to become other cell types when other cells in the embryo have lost the ability to do so. So I really kind of started to build a bridge between embryonic stem cells and neural crest cells. And those findings opened my mind a little bit more, and I'm trying to understand really how do cells make developmental decisions from really early in development. And as a postdoctoral scholar at the California Institute of Technology, I really wanted to use the techniques that were developed at Lone Chi Lab. So I was a developmental biologist coming to a technology lab to ask some really deep developmental biology questions. And currently, I'm quantitatively studying the distribution of mRNA during early mammalian embryogenesis using humans and mouse as a model systems. I was fortunate that during my first year, I was named a Hannah Gray Fellow. That always speaks about my commitment to DEI and seeing to inspire underrepresented scientists through my work. Some of the DEI activities I really want to be part of, like here in UCSD, is to actively participate in faculty and graduate students' process, advocate for equal opportunities for funding and laboratory space for everyone, and create programs for students and postdocs to allocate funds that pay for childcare. I'm a mom, and I was a mom as a postdoc, and I really know how important this is for avoiding the leaky pipe that we have right now in the system. And I want to foster earliest interest in STEM in young kids, because I think just by planting the seed really early in development makes a difference. And this is some work that I have been done since I was a PhD student at Northwestern University. So now I'm going to talk about the hardcore science. So during early development, the mammalian embryo sequentially generates different derivatives and different lineages. So we all know that the totipotent, the fertilized egg is totipotent, meaning that it can give rise to any cell type in the organism. As development progresses, this potency is lost, and then the embryo has to make different developmental decisions. The first one, which will be the first part of my talk, is whether to become pluripotent cell or form extraembryonic tissue that is going to form the placenta. And from pluripotent cells, we know that all cells in the embryo are going to be formed. The second part of my talk is going to be focused on understanding how cells go from pluripotent cells to actually leading to the foundational lineages that are going to form the embryo, that are the ectoderm, mesoderm, and endoderm. And from these lineages, all the cells in the embryo are going to be formed. And why do we care about this? Totipotent cells could allow to deriving all the extraembryonic and embryonic phase of the developing embryo. So we are now facing an era that we have a lot of models to studying human development or mammalian development in vitro. But these models lack some of the cell fates that are created in the natural embryos. I am interested to know what transcription factors and signaling inputs are required for the totipotency of cells to be maintained, and what destabilize their totipotent state and induce differentiation. And I want to do this because early human development frequently fails. Naturally, as we were discussing with Catriona recently in our talk, like many pregnancies are fail between the first trimester of development. And by understanding how normal development occurs, we can understand what's happening in these early stages of development. And we can potentially bring light to have more tests of IVF embryos that could be successfully implanted into moms. So I'm really interested to understand how do individual totipotent cells in early mammalian embryo begin to differentiate and when. And I use mouse embryos as a model system to study this question. And my hypothesis is that really totipotent cells maybe are not as equal as we thought. And maybe a special temporal distribution of mRNAs within and between cells in the same embryo could give rise to the differentiation of different cells. So I use mouse embryos for multiple reasons. First is that they are transparent and they are small. And second, because the first main developmental decisions happen only in 4 days of development. So from zygote to 2 cells, you have the maternal to zygotic transition. So transcription starts to happen around this time. And then between 8 and 16 cells, we think that the first cell fate decision occurs. And this cell fate decision is between either the inner cell mass cell, which is depicted here in yellow, that will form the pluripotent cells, and then we call it the inner cell mass cell. From this group of cells, all the cells in the embryo are going to form. Or become trophectodermal cell, which is depicted here in blue, that will form the placenta that will be important to attach to the mom, to form the placenta that is going to be attached to the mom. So these kind of questions has been asking in developmental biology in the field for a long time. Some single-cell transcriptomics methods has been used to reconstruct these developmental trajectories, but these methods involves cell dissociation of whole embryos, sequencing, and reconstruction of development. And despite they are very powerful and then allows us to know which molecules are present at given time, and then in the abundance of them, these methods ignore spatial distribution within and between cells that I think are going to be key to understand many developmental processes that happen that early in development. And then also it's important because complex developing tissues have unique RNA and protein expression patterns in cells that are located in different places and positions. So for example, here we can see early mouse embryo in which the red protein is marking for pluripotent cells that are expressed only in one pole of the embryo, and then the green protein is marking the trophectodermal cells that is kind of surrounding these pluripotent cells. And also we can see here in human gastruloid that there are gastruloids that are concentric rings that are especially organized into different cell fates, and the position of the cells and then what they are expressing is really important to understand how development occurs. So when I arrived to the Kai lab, I was amazed by the techniques that they developed. So all the techniques that we use are based in something that we call single molecule fluorescence in situ hybridization, and then we call smFISH. Using smFISH, we can map mRNA transcripts in space in high fidelity. We base the techniques in the development of that was made in 2006 by Arjun Raj when he was at MIT, under Van den Arden's guidance. And then he has this clever idea to design multiple probes against the same species of mRNA that were fluorescently labeled. So when you image the sample that was previously hybridized, you can see single dots that represent a single molecule in this embryonic cell. And this is an example of mouse embryonic stem cells. So single molecule FISH allows us to detect individual mRNA molecules in each cell. So we can see, for example, here expression of two different mRNAs. The names doesn't matter for now, but we can see not only how much expression do they have, but also where in the cells these mRNAs are actually present. So the first experiments I did when I arrived to the Kai lab were to actually adapt single molecule FISH to mouse embryos. And then this is a housekeeping gene that we call that is called EF2. And we can see that is homogeneously expressed throughout the embryo. This is a 4-cell embryo. And then the way that we assess RNA quality is really simple. We design probes against the same mRNA species, 2 sets of probes. One of the sets have one fluorophore and the second another fluorophore. When you do the imaging and colocalize the images, if the colocalization is perfect, you can say that this mRNA is intact. So when you have this, you are confident on the data that you are getting out different readouts. But I faced a huge problem when I was doing these experiments is that until now, most of the experiments that were done doing special transcriptomics or using SeqFISH were done in a monolayer of either cell culture or tissue section. And I wanted to map mRNA molecules throughout the embryo that is 50 to 70 microns thick. So I had to combine clearing protocols and adapt them to embryos that they are very, very fragile, embryos and human gastruloid colonies, and actually work with different settings of the microscope to be able to map with high fidelity single molecules throughout the whole embryo. So when I had that developed, we started to do sequential fluorescence in situ hybridization that we call SeqFISH. And the way it works is also very simple. So we designed mRNA probes against mRNAs of interest. So we designed between 25 to 30 probes per species. And then what is different from the original single molecule FISH is that the secondary readouts are chemically designed to be able to strip off after the secondary hybridization. So in this way, like for example, if you have 3 channels in the microscope, you can image mRNA 1, 2, and 3. and strip them off, and then in the second hybridization, just hybridize mRNA 4, 5, and 6, and strip off as many times as you want until you get the mRNAs of interest. And we can do this because we have amazing engineers in the lab that design a robot that is doing the work for you. So basically, you hybridize primary probes at the same time, and the secondary probes and washes are made with this confocal imaging and microfluidics automated system that enable this sequential imaging over time. So you can do, for example, place the sample in the afternoon and then have the microscope running throughout the night, and the next day you come for the data. And this is how it looks like. So this is a 4-cell mouse embryo. We're looking at the expression of EF2, and here we're seeing that with high fidelity I can map mRNA at the single molecule resolution really well in a thick tissue. And here, when we strip off and hybridize the first readouts, this is how it looks like. So we can see clearly that the first 3 mRNAs are being expressed in this image. So after getting the data, we had to develop a pipeline for studying these quantitative, being able to quantify the molecules and being able to do some systems biology approaches to understand embryonic development. So in collaboration with Nicolas Pelaez from the Elowitz lab, we developed an automated nuclear and membrane segmentation first that will allow us to look at create objects like a mask that we will be able to detect the single molecules in each C. This is just a small example that I have here, how we do it. And we can just do downstream data analysis. So this demonstrates for first time that we can not only detect single molecules in a whole mouse embryo, but also that we can do quantitative analysis. And this is an example of EF2, which we demonstrate that both of the sister cells have similar spot intensity and distributions and quantification in both of the cells. So this is kind of my proof of principle that the technique is working. So now when we had that, we started to ask interesting questions like, do different cells in the early mouse embryo differentially express mRNAs before the specification of this cell fate? So in the field, it's been known that the first cell fate decision in mouse happens between 8 and 16 cell stage. And as I told you before, this decision is between becoming pluripotent cell or inner cell mass cell or trophectoderm that is going to form the placenta. Currently, there are 2 models that explain how this decision is made. The first is the polarity model that really like the differential expression of the proteins upon cell division will generate a cell that have 2 different proteins that will generate trophectodermal cells, and cells that have only expression of 1 protein that is called apolar will form inner cell mass cell. And the second model is the positional model, which stands for is the position of the cells within the embryo that is going to be important and critical to be either inner cell mass cell or trophectodermal cell. If my hypothesis is correct, we will find early asymmetric mRNA distributions early on in development within single cells in the same embryo that could potentially give rise to subsequent differential RNA expression that will lead to future cell fate choices. So I decided to do a pool of about 200 mRNAs for my first experiment. And then I included, of course, mRNAs encoding for proteins that are important for supporting either the positional or the polar model. So what I found is, for example, in CoA5, as we can see here, is homogeneously expressed in the 4 cells of this mouse embryo. And this in CoA5 is described as one of the key proteins that is important for the polarization model. In contrary, mRNAs such SOX21, we found that is differentially expressed already at the 4-cell stage. And then it's largely known that SOX21 is marking the presumptive pluripotent cells. So I wanted to look also at SOX2, that is a pluripotency marker. And SOX2 is really lowly expressed at these stages of development. But one of the advantages of SeqFISH is that we can actually detect the abundance of SOX2 or low transcripts, which show us that SOX2 is also differentially expressed at the 4-cell embryo, as we can see here. And interestingly, SOX2 is expressed in the same cells that SOX21 is expressed. So we did the same analysis with many molecules, and we found that already at the 4-cell stage, maybe the cells are more different than we thought. And maybe totipotent cells at this stage are not the cells are not actually totipotent, and they are already making decisions to become pluripotent cells. So with this SeqFISH, we wanted to quantify these results. So what we found is that mRNAs that were important for supporting the polarization model are actually uniformly expressed in the cells. This is the control EF2 showing equal expression of the molecules in all the cells. What we can see here is that we can map the mRNA abundance of the cells, and we can see here, for example, in SOX21, that these two cells are differentially expressed the mRNA compared to other cells in the embryo. And we corroborate this also with SOX2 and other molecules. So once we have all this pool of data, we started to do some clustering analysis and principal component analysis to see whether or not the cells at the 4-cell stage embryo are already differentially expressed some mRNAs. And we saw that these two cells, yellow cells over here, are more similar to each other than the other cells in the embryo. And when we did the principal component analysis, it corroborated that cell number 12 and 10 are more similar to each other than other cells in the embryo. So we had this data for other stages of development, and then for patent reasons, I can't talk about these results, but they are going to be published hopefully very soon. So this data that we have right now suggests us that the symmetry of the embryo is broken before we thought, and maybe that the first cell fate decision happens already at the 4-cell stage because of the differential expression and transcriptional signatures that these cells have. Led us to hypothesize that maybe the cells at this 4-cell stage are already having some level of differentiation. So we started to ask more interesting questions, like not only comparing cells to cells and what is present here and there, but also about the mechanisms that are probably generating these early asymmetries of mRNA abundance between sister blastomeres during early mammalian embryogenesis. And I hypothesized that maybe oscillations of global transcription could be a mechanism by which early asymmetric RNA distribution give rise to this first cell fate decision as early as 4-cell stage. So to test this hypothesis, I decided to generate a slightly different probes. So I designed probes against the intronic regions, and I randomly chosen 1,000 mRNAs, and I designed the probe that they all have the same secondary readout. So in one snapshot, I can see global instantaneous transcription activity that is happening at that particular time in the embryo. So when I did the first experiment in an 8-cell embryo, I saw a really nice result that shows us that not all the cells in the embryo are actually expressing the introns, and that there is potentially a differential global instantaneous transcription that is happening between blastomeres already at the 8-cell mouse embryo. When I did the same experiments using 4-cell embryos, I found that not all the cells in the embryos present the same global instantaneous transcription, suggesting that the blastomeres at the 4-cell stage already have this differential global instantaneous transcriptional activity. So I wanted to multiplex the different techniques and look not only at the introns, which is the pre-mRNA, but also the mRNA in the same embryo, and then trying to correlate between instant transcription and mRNA distribution. And then one of the genes that I thought that was going to be very important to see at these stages of development was HES1, because we know that HES1 is part of the Notch signaling pathway and is important for many, many developmental processes. But we study them in different contexts, such neurogenesis later in development, and also somatogenesis. So I asked whether or not HES1 is first present, and second, if it's present, differentially expressed in the embryo at the 8-cell stage. To my surprise, HES1 is not only present, but is present and is differentially expressed. And we think that the cells that have more intron expression that I'm showing here with these arrows are the same cells that have more expression of mRNA. We can see this more clearly when we quantify. So here, the small dots represent introns, and the big dots represent mRNA, in this case, HES1 mRNA. And then we can see that the cells that have more introns also have more mRNA associated to those cells. And I wanted to go further and see other layers of regulation. So maybe if HES1 is there, I wanted to see also if the protein, like the mRNA and introns are there, I wanted to see if the protein was present. And then when I did the experiment, looking at the protein, I saw that the HES1 is present at in early embryogenesis and is also having differential expression in the cells in the same embryo, suggesting that maybe this oscillatory behavior of transcription is not as crazy as we thought at the beginning. So I wanted then to multiplex 3D SeqFISH that allows the visualization not only to the mRNA, but also to the introns, like the pre-mRNA, but to the mRNA and the proteins. And I was managed to design the secondary probe to visualize proteins using the same principles that we use for mRNA, so we can now see a couple of proteins in the same sample. And this is how it looks like. So this is the pre-mRNA at 4-cell embryo, looking at the expression of introns. And this is the mRNA looking at the intact mRNA expression of EEF2. And these are how different proteins look like in the same embryo. So we have this membrane marker and also the transcription factor of interest that in this case is HES1, showing heterogeneous expression within the cells. So as conclusions for this part, I have that 3D SeqFISH was developed to image introns, mRNA, and proteins in whole month pre-implantation embryos, preserving spatial distribution of molecules and cells within the sample. That our computational pipeline allows 3D segmentation and single molecule detection of mRNAs and quantification of spatial distribution of the molecules within the same cells of intact mouse embryos. And that 3D SeqFISH reveals differential mRNA expression of different factors that suggest that maybe the blastomeres that already at the 4-cell stage are different and not as we thought. And that differences in global transcription detected by measuring introns and HES1 mRNA and proteins at 4 and 8-cell mouse embryos suggest that there could be an early cell fate specification that we could precede by transcriptional oscillations. So in the second part of my talk, I'm gonna focus on early human development. So I just talk about how totipotent cells could potentially differentiate between pluripotent and extraembryonic tissues. And now I'm going to focus on how pluripotent cells can just how from the signaling molecule pathways, the pluripotent cells are actually becoming either ectoderm, endoderm, mesoderm, or progenitors of the germlines. And then I'm using human gastruloid colonies to create multiple embryonic and extraembryonic fates that are especially organized. And the way that we do it is really cool. We use induced pluripotent stem cells as small colonies that we induce using BMP4 ligand to form these different derivatives. And what is very beautiful about this system is that we now can have access to more human samples to be able to study developmental biology in humans. And then after 48 hours of differentiation, the cells self-organize in these concentric rings that are representing cells from different germ layers. For example, the extraembryonic tissue depicted here in green and here. The mesodermal derivatives depicted here in yellow and here in green in the protein level. And then the core of the colony is representing the ectodermal lineages over here, showing here in blue. What is known about development of human and mammals and many other organisms is that paracrine signals are involved in this process to make like the cells different. And paracrine signals are signals that have a source and diffuse and create gradients, and morphogen concentrations are different, and this represents the French flag model that creates differential expression of transcripts that are going to form different cell types. So Wnt, BMP, and Nodal are widely studied during development. And it's believed that BMP4 induces BMP, BMPs induce Wnt, and Wnt induce Nodal. And this cascade and circuit is in charge of generating all the differences between the cells during development. And we ask whether or not juxtacrine signals that are cell-cell contact signals are actually important for this human development. So Notch was our best candidate to study the juxtacrine signals in the human context. So we use a systems biology approach to investigate how Notch signals generate gastruloid colony differentiation. So we use transcriptome analysis using single-cell RNA-seq in collaboration with Matt Thompson's lab. We did a special expression of the Notch components. And also we did some immunofluorescence-based special protein expression analysis at single nuclei resolution that allows us to not only look at the expression, but also quantify and put numbers into our hypothesis. So we hypothesize that if Notch plays a role in early human development, the components necessary to transduce Notch signals should be expressed at the right time and the right place during human gastruloid differentiation. So when we did single-cell RNA-seq, after 48 hours of differentiation, we found 8 clusters that represent all the cell types that we currently find in gastrulation of human embryo. And those are the ectoderm, germline over here, the mesoderm, represented here by mesoderm 2 and mesoderm 1, this is going to be important later in my talk. Endodermal cluster, and we also look at the extraembryonic tissue over here in this part of the cluster, and then the epi-like cells that are part of the epiblast. So when we look at the Notch components, we saw that the Notch receptors are actually present in the gastruloid colony after 48 hours of differentiation. So Notch 1, Notch 2, and Notch 3 are present. We also look at receptors and direct transcriptional targets, and we found that HES1, DLL3, and JAK1 are expressed. Importantly, DLL3 is expressed only in the mesoderm, and JAK1 is expressed mainly in the ectodermal area and also some of the ectodermal area and some of the extraembryonic tissue. So not only they are present, but they have some sort of a special distribution. Some of the direct targets from the HES and HEY family are also present, and then also have some special distribution that could be interesting to understand. So in order to see these special differences and whether or not Notch have a combinatorial potential code that is giving the cells, like acting as like this combinatorial code can be acting to be able to regulate when one cell fate ends and then when the other one starts, which was our hypothesis. We needed to see the special distribution of these Notch components in the gastruloid colony differentiation. So we did SeqFISH against the whole Notch signalome. We used the same approach using the multiple primary probes against the mRNAs of interest. We look at the single molecule FISH of all of the molecules at the same time in the same sample, as we're looking here in this video. We did some 3D segmentation and identification of the molecules in the tissue and some quantifications. The first corroboration that we wanted to have to continue further analysis was whether or not protein and mRNA expression was correlated. So we look at some fate markers such HAN1, depicted here in red, that is corroborating with the HAN1 expression over here, the protein expression. TBX2, TBXT, that is expressing in the mesoderm, looking at the expression here correlating with the green expression over here, like the protein. And then also the SOX2, which is marking the ectoderm epiblast cell population, correlates with the protein expression. So we can confidently say that mRNA and protein expression is actually matching, and we can just do a special analysis of the different components that are part of the Notch signaling pathway. We include ubiquitin ligases, like some of the core machinery proteins and components. We look at the ligands, we look at the receptors, and then transcriptional targets. Interesting, what we found, not only using SeqFISH, but also using single-cell RNA-seq, is that there are two compartments in the mesoderm that are going to be very key for how we think Notch is acting during gastruloid colony differentiation. So when we looked at the Notch ligands, we saw that there is a special distribution of JAK1 that is mostly expressed at mesoderm 1 over here compared to mesoderm 2. And also when we saw expression of DLL1, it was mostly expressed in the mesoderm 1 compartment compared to the mesoderm 2. And this is corroborated by also the single-cell RNA-seq. What was interesting is that not all the components that we found using SeqFISH were present during the single-cell RNA-seq, because sometimes single-cell RNA-seq drop off very low expression levels that we can actually rescue using SeqFISH. So both of the techniques are actually important to be able to see where the molecules are expressed and how and the abundance of the molecules in the tissue. When we compare the ligands with the receptors, we also found that they could be a potential combinatorial code that is important for understanding Notch during human embryonic development. So we found that Notch3 is actually surprisingly expressed mostly at the mesoderm 1 compared to mesoderm 2. And then also, when we look at the targets, we see that, for example, HES4 is mostly expressed at mesoderm 1 compared to mesoderm 2, suggesting that this subcellular compartment localization of different components of Notch will be very important to understand how Notch acts during gastruloid colony differentiation. So after this, having these findings, we wanted to know if Notch signaling is actually required for human gastruloid cell differentiation. And then our hypothesis, after finding a special distribution of molecules and then single-cell RNA-seq, we thought that maybe Notch was important for mesoderm formation. And when we block Notch signaling using small molecules, DAPT and LY, we found that TBXT expression was affected, and some of the expression was gone in some of the compartments of the TBXT here if we compare with the control. Interestingly, the endodermal cell fates are also almost completely gone. When we see the expression of SOC17 over here, we can see high expression of this marker in some of the cells in the colony, but after inhibition of Notch, we can see that these cells are almost completely gone. And this happens at the expenses of the SOX2 expansion and also HAN1. So we think that Notch is acting as surveillance system to be able to say, like, this is the mesoderm, so mesodermal cells start from here to here, and then the rest of the cell fates are going to be actually surrounding this domain. So we wanted to do some quantitative analysis on this. So we developed a way to be able to quantify proteins at single molecule, single nucleus resolution. So we can see here expression of SOX2, TBXT, and HAN1. So TBXT is here is the segmentation and quantification of TBXT, for example. And we can look at the amplitude of TBXT, shown here in the red curve, that we can see that when TBXT start to be really high regulated in the colony is when the SOX2 cells start to go down. And then we can see that where the expression of HAN1 starts to be upregulated. And then when we do the blocking Notch signaling, we can see that the amplitude of SOX2 is going down at the expenses of either ectoderm or trophectodermal cell fates, which means that Notch regulates the local amplitude of expression and the position of fate boundaries in human gastruloid colonies. So as a summary, in this second part of the talk, I can say that Notch signaling components are expressed during human gastruloid colony differentiation, that the expression of Notch components discriminates compartments within individual germ layers, for example, the mesoderm 1 and 2, and that Notch activity is required for mesoderm and SOC17-derived cells populations that are deriving endoderm and potentially primordial germ progenitors cells. And that Notch regulates spatial patterning and the relative proportion of cells allocated to different fates. And Notch signaling regulates the amplitude of expression of lineage-specific transcription factors that control this gastruloid colony differentiation. So we can map a model in which not only paracrine signals are important for human embryonic development, but also juxtacrine signals are acting as a major player during human gastruloid colony differentiation. And as of conclusions of my seminar, I have that we developed this 3D SeqFISH method that allows us to measure RNA expression at single molecule resolution. especially in intact tissues, and can be multiplexed not only using RNAs, but introns and proteins. That early mouse embryo blastomeres exhibit differential RNA expression at the 4-cell stage. That could lead us to describe a new principle in developmental biology in which the cells are actually acquiring cell fate specification earlier than we thought, and that differentiation of human germ layers combines paracrine and juxtacrine signals and requires notch signaling in human gastruloid colonies. Now, in the future directions and vision of the Buitrago-Delgado lab, I want to continue investigating the mechanisms that mouse and human stem cells use to make developmental decisions during early embryogenesis. So I want to create comparative maps, transcriptome-wide spatial temporal RNA expression atlas in the early human and mouse embryos from fertilization to blastula stages, to be able to compare evolutionary similarities and difference between these two species. Here we can see a blastula stage human embryo that I luckily had access to, and I apply single molecule FISH looking at EF2 to see RNA integrity, and we are confident that we can do the experiments in human embryos the same way that, or similar ways that we do in human embryos. And I want to do this by using SeqFISH Plus that was developed in the lab that enables measuring the transcriptome of each cell, basically. And we use unique temporal barcodes that can unambiguously identify thousands of mRNAs in situ that can map more than 1,000 mRNAs in only few rounds of hybridization. So for example, if you have a barcode that uses 5 different fluorophores in 6 rounds of hybridization, you can get up to 15,000 mRNAs and map in, and then you don't need like to do the sequential FISH, which will be impossible to do with that many genes. And I'm very interested to continue investigating the mechanisms that they generate this RNA expression, the asymmetric RNA expression during early mammalian embryogenesis. And I want to combine live imaging, like for example, using MS2 systems in mouse embryos to be able to map the transcriptional activity of the cells, whether there is a pulse that that is unique to generate, a unique pulse that can generate differences, so there is an oscillating behavior that is occurring in the cells in the same embryo that is actually giving us the differential expression of transcripts. And I want to combine this with SeqFISH to be able to tease apart the mechanisms that actually generate these asymmetries early on in development. And finally, I want to study dynamics of signaling pathway, and I want to combine using reporter lines that we already have in hand to be able to build human blastoids that I'm showing here in these pictures that are kind of the next stage after losing pluripotency, is to form like these blastoid that comprised by primitive endoderm, pluripotent cells, and trophectoderm. And by looking at the dynamic expression of different signaling molecules could be important to understand how this early human development works. And with this, I want to thank everyone in my lab that has been part of the project. This is a multi-lab collaboration between Kai Lab, Elowitz's lab, and Matt Thompson's lab. Carlos Lois has been really important for input in Notch, in the Notch project. And then I also want to thank Nicolas Peláez, who has been a really important part of both of the projects. And with that, I want to take questions.
So we've got some time for some questions. If you want the mic, just raise your hand. I'll start with a question while people are getting together. So when you see the differences in mRNA between the cells at the 4-cell embryo stage. How reproducible are those? Like, are you always seeing like one cell looks like this and one cell looks like that, or is there a stochastic nature to it?
That's a really great question. For SOX21 expression, I consistently see repetitively in different embryos that there is a expression of like in 2 cells more than other cells in the embryo. And when I corroborate with SOX2, same cells are expressing more SOX2 than the other cells in the embryo. I repeat these experiments already a couple of times, and I get the same results. So I'm confident that this is a real result.
Yeah, that was brilliant. I did have a question about shear stress and flow. So when George Daly and others published a paper on mouse embryonic stem cell development and the cell fate specification, they talked about having to have shear stress, flow, and then, obviously others have worked on stiffness of the matrix. Have you thought about putting these in a more 3-dimensional matrix and adding flow to your system? Because it may change the outcome and you have a perfect system to look at that.
Yeah, no, that's a great suggestion. I haven't think about that, but that could be very doable in the near future. Thank you.
Hi, I have a quick question. So actually I have 2 questions. So one is more on the biology of it. It's beautiful talk, amazing image. I've never seen anything this amazing. So the first part is, as notch pathway goes, do you know which particular notch is important? Notch 1, 2, or 3, or do you see a difference there?
That's a really good question. We did siRNA. I think I have the question, the slide here. We did siRNA against the notch 1, 2, and 3. And it seems like notch 3 is really important for it is the receptor that is important for the mesoderm formation in this case. So we were very worried that maybe only using small molecule inhibitors, we will have effects on other things, because we know that gamma secretase acts in like super different contexts. So we wanted to go further and did siRNA, and we found that notch 3 is the one who prevents normal mesodermal differentiation of the mesodermal differentiation. So we think that notch 3 is the player that is important here, which is really interesting, because if you know mouse literature, in mouse literature is notch 1 and 2 that are important, but the more we study human development, the more we learn that humans are very different. So that's why I want to start working more on human development and understanding and learning more about the real embryo and the real in the real context.
Okay, that's fascinating. So my second question is a little bit more on the technical aspect of things. So there are a lot of like pediatric cancers that's been showing that mutation, for example, can occur really early at embryonic stage, for example. I'm wondering if we can use your technique, or if you think your technique can be applied to detection early mutations, for example, using the FISH method.
Yeah, that's something that we want to do in the future in my lab. Right now, it took a long time to develop the pipeline, the techniques, and now it's like the exciting time to start to do some genetic and CRISPR probably knockouts, and then see the effects of this in embryonic development. Thank you.
Hi, we're here. Beautiful talk. So that's really fascinating to me, because, in the context of the embryonic stem cells, they basically form their own environment, their own niche in a way. When you show that at the 4-stage cells, there's already kind of state specification, have you thought maybe to kind of take those cells with the low mRNA to put them in the context of other cells that actually have how plastic this system is? And I think from the perspective of, do they maintain some type of a memory that they were previously kind of low mRNA on that particular state, and your thoughts about that?
Yeah, thank you for asking the question. We've been thinking about these experiments, dissociating the cells and choosing the cells that are more pluripotent-like cells than other cells. We are setting up the the system to be able to do it in the lab. Just to remind you, when I arrived to the lab, there were not even animal protocols to work with, so I had to start from zero. And then now we're just becoming more and more developmental biology, and then we want to do these kind of experiments before I leave. Yeah, thank you.
So if there are no other questions, let's thank Dr. Buitrago-Delgado again for a terrific talk.